So as I complete the step, I'm also writing it in my notebook. And if we were to have someone from the FDA come in or someone to audit us, and they see that I'm not doing that correctly, we can get in trouble. So it's just a lot more regulated of an environment. So before I get into testing, can you guys tell me some of the projects you guys are working on? So I kind of know how to or ideas for projects, which you want to do. Go ahead.
I already told you mine, but I'm working with cell cultures and I'm subjecting them to urushiol, which is the oil inside poison ivy, and I'm seeing if it kills them or what it does to them, because it has the power to break DNA.
Right, so you would be doing more of a cytotoxicity assay. So how many cells are alive versus how many have died in your plates, right? Okay, so do you know what type of cells you're looking to use?
I'm using Dictyostelium, mammalian, and E. coli.
Okay, so all different. So I work a lot with mammalian cells. A lot of the cells we use mimic human cells because I work in pharmaceuticals, so a lot of the drugs we're developing or other companies are developing that we're doing testing for are for mammalian cells, of course, because they're for humans. So with your test, you would basically, have you started doing stuff, or are you, is it just an idea?
Just learning how to culture.
Oh, okay. All right, so just learning how to culture cells. So that is actually one of the first steps that we teach our new analysts. So you can actually come to Lancaster Labs directly out of college with no experience, which is kind of nice because Lancaster Labs is willing to teach you everything you need to know. And if I learned anything in college, it was how little I did know when I went into industry, which is completely fine. But with culturing cells, you're having to use aseptic technique, correct? So do you guys, can you walk me through your aseptic technique here?
Not really, but you get a Rini, what's this? She wanted to know what the technique was for culturing cells.
So right now we're starting with Dept cells.
Okay.
So I've been showing them how to passage them, so they've been scraping it with
Okay.
using the aspirator.
Okay, so you never actually, do you remove the cells from the plate and then put them in a new flask or plate? Okay, all right, so that's basically how ours works. Do you guys use aseptic technique? Do you guys work in a BSc?
Yeah, we have a biosafety.
Okay, so you
We
have
have her a little tour that I have to rename. We got 2 biosafety level 2 cabinets now.
That's awesome. Yeah, that is so cool. Okay, so we have 12 biosafety cabinets in our department alone. And our technique is so every technique that we follow is written out in a method. And we have to follow stepwise what that method says. So with I don't know if you guys do this, but we actually have to sterile glove and wear sterile sleeves. Do you guys do that?
We do the sterile glove.
Okay, you don't wear the sleeves that come up your lab coat sleeves? Okay, so we have that extra protection there of the sterile sleeves. And then we wear the sterile gloves. Do you guys spray your hands down with IPA or anything like that? Alcohol? Ethanol? Okay, yeah, so that technique's actually what we use in industry as well, except we use isopropyl alcohol. And we have to clean, do you guys clean your VSCs before you do anything with cell culture? Yeah, okay, so we use something called spore cleanse in our VSCs, and they have to be sprayed down completely and dry for 15 minutes before we're allowed to sterilely enter that VSC. And then once you're going in and out, say you have to bring your cells into your VSC, are you guys spraying those down, like your plates and things with IPA? Yeah, good, awesome. So that would be basically our aseptic technique training at Lancaster Labs, is basically teaching people how to do that. And then do you guys make sterile medias for your cells? Yep. So then the next step in our training process is making those sterile medias, and then they have to incubate in a incubator for a week and make sure they don't grow anything prior to us being able to say, okay, person can aseptically go on MBSC, and we can trust that they're not going to contaminate anything. And then we move on to cell dosage. So our employees learn about adherent lines and suspension lines. Are you guys familiar with those? Some of you not. So an adherent line is a line of cells that sticks to the plate. So they grow on the back of the plate or the back of the flask, and then they have to be either scraped off the back of the flask, or you can use something like trypsin. Do you guys know what that is? Okay, so you guys trypsinize your cells. Oh, you do? Okay. So trypsin or acetase can be used to break the barrier between the cells in the flask. And then they have to be rinsed, and they go through a process of spinning them down and isolating the cells. And then do you guys count your cells to see how many cells you actually have? Yes? Okay. So then you count those cells, and then you figure out the density that you're needing for your experiment, or the density you just need to keep those cells alive, right? So as far as your experiment goes, using the liquid from poison ivy or whatever you were saying is, so on mammalian cells, the way we would test something like that is we would seed 96 well plates with, do you guys know what those are? Yeah? Okay, sorry, I'm not too familiar with what you guys are doing and what you aren't. So we would use a 96 well plate, and we would seed our cells directly into that plate. So instead of putting them in a flask or something like that, we would put our cells directly into those 96 wells. And then we either do drug dilutions. So in your case, you could easily dilute to see at what concentration that substance is going to destroy that DNA and kill the cells, or just put it directly on them. But I would dilute them. So we dilute all our drugs. A lot of our drugs are used for potency studies, so we want to see at what potency you are able to kill those cells or produce the growth factor that you want or anything like that. Does that make sense? So in your study, dilute that and then add that to your cells that are in your plate. Either you can do it the same day or you can do it the day after. It just depends on the assay. With us, I have some testing that I seed my plates the day before. I let them incubate overnight and grow up a little bit, and then I add directly to that plate. That's what you were thinking?
Yeah.
Yeah, that's awesome. And then you can use something that either goes into the cell. So I only run one cytotoxicity assay, and we use rizazirin dye that goes into dead cells, and it causes a reaction in those dead cells, which causes a fluorescent signal. So then you get a gradient across your plate to see what cells are dead or what cells are alive. Yeah?
Yep.
So that's pretty cool. Does anyone else have any projects that they're working on? We're excited about ideas? No? Do you want me to just talk about other testing I do? Yeah? That works? Okay, so do you guys know what ELISAs are? So ELISAs measure protein concentration or antigen-antibody connection. So an ELISA is something you can do in a wet lab. You don't have to do it sterilely, so that's kind of nice. Those usually start with a coating antigen, and then you let it incubate overnight. And then the next day I would add my drug to that, and I would see how much of that drug is able to bind to that antigen. So the drug, usually in an ELISA, what we're looking for is, say there's some antibody growth factor, something that's on a cell surface of, say, a cancer cell. I'll make one up for you guys. So on the surface of a cancer cell, and if we inhibit that ability to be expressed on that cancer cell, we're going to kill the cancer cell, because that's the only way that we're going to be able to make the cancer cells continue to proliferate and continue to grow. So what I would do is take that antibody or antigen or whatever's growth factor, whatever's being exposed exposed on the cell surface, and I would bind that to a 96 well plate. And then I would take my client's drug and I would put it on the cell, or put it on the plate that's already been coated. And we can measure, and once again diluting that. So we have different dilutions, and we're able to measure at what dilutions you are, like what's the best dilution of drug to use on those samples. Because here we have an overabundance of extra that's not binding, or over here we have not enough, we still have cancer cells growing. Does that make sense? So that's kind of what an ELISA does. And in an ELISA, you bind your drug, and then you bind another antibody to that fluoresces, and you're able to measure, you're able to measure and absorb its reading. And that's how you get your points used there. Do you guys know what gel electrophoresis is? No? Okay, so that's another protein test we do. So gel electrophoresis actually allows you to see, I'm trying to think of a good example. You can measure different segments of DNA. So this would actually potentially work if you could isolate your DNA in some way. I don't know if you guys do PCR experiments that you would be able to isolate your DNA. And then, so PCR amplifies a certain segment of DNA that you're looking for. But with yours, you're kind of looking to kill the DNA.
Yeah.
But then you can, if you're not doing real-time PCR, which does the quantifying for you of those DNA segments, you can actually run it out on a gel and see if the band of DNA that you wanted to produce actually showed up in your samples. So that's kind of cool. And then my biggest project at Lancaster Labs since I started is I'm working on a mycoplasma detection test. So I've been with this project since the very beginning. I jumped on it about a year and a half ago. So mycoplasma is a type of bacteria that doesn't have a cell wall. And if you guys know, do you guys know anything about antibiotics and how they work? Yeah. So a lot of antibiotics target the cell wall to destroy a bacteria, right? So mycoplasma doesn't have a cell wall, and therefore a lot of antibiotics don't target it. So they become prevalent in a lot of cell lines because nothing can kill them. So in our cell culture, I don't know about in your medias, we have antibiotics in our medias to make sure that our cell lines don't get infected. Does that sound familiar? Yeah. So with mycoplasma, the antibiotics that we use, like penicillin, it won't affect a mycoplasma species. So there's other departments at Lancaster Labs that we have a department called cell banking. So all they do is grow up cell lines, and they produce cell lines that clients need for specific testing that mimic cancer cells or mimic a different disease cell. And those cell lines have to go through a mycoplasma detection test to make sure that there's not bacteria in those cell lines that don't have a cell wall. So my project has been working with different cell lines that are infected with mycoplasma and seeing at what is the lowest concentration we can detect mycoplasma at. So in this process, we went through a feasibility study, and that feasibility study encompassed months and months of random testing of just, here's the cell line, see if we can produce, if there's mycoplasma in it. Let's add mycoplasma to that sample and see if that cell line still produces, if you can detect the mycoplasma that you know you added. And all sorts of things to mitigate interference. So sometimes the medias that the cells are kept in can somehow block the PCR from occurring. So from that DNA of the mycoplasma to amplify, it can be blocked by different things in the media. So we have to do interference testing on those samples to make sure that the mycoplasma can be detected in that media. So that's all encompassed in our feasibility studies. And then we move into validation. And validation is basically proving a point. So you're proving that this will work 100% of the time. You will always be able to detect mycoplasma. And that results in multiple analysts running it so that you have, it's not just one single analyst being able to prove that there's mycoplasma. So you have to have multiple analysts, multiple runs, and you have to use multiple different types of mycoplasma. So we spike with 3 different kinds of mycoplasma, one at 2 different concentrations. And then we have to produce the results we're expecting. So basically like how you guys would run a study here. But then we have to write up a report, and the client has to sign off on that. And we then are able to do routine testing for them. So they send in samples, and they say, okay, we need this cell line tested for mycoplasma. Can you do that for us? So that's been the big project I am working on. It is, so the way we detect the mycoplasma is using PCR. First, we have to separate the cells from the media, and we actually process the 2 separately. And then we go through a couple steps to get rid of any, well, we use an RNase digestion and a DNase digestion. And then we recombine the supernatant that has no cells in it and the cell pellet, and we extract that. Actually, you guys don't have a ton of experience with PCR, right? So in the PCR that you guys would do here, you would be using probably column extractions. So basically you just centrifuge things very quickly to separate based on density. We actually have a machine that we use for mycoplasma called Magmax, and it actually uses magnetic particles to separate the DNA from anything else that's in there. So we extract our DNA, and then we amplify it using PCR, and then we get a result that's either positive or negative. And based on, if we're running routine samples, we want that to be negative because you don't want mycoplasma in your samples. If we're running a validation and we've spiked that with live myco, we want it to be positive. So, do you guys have any questions for me? No? Any other projects you guys want to talk about? What grade are you, most of you in? Okay. You're going to be juniors? Awesome. Great. Oh, 10. Sorry. Awesome. So you guys have a lot of time to do something. Do you guys know, like, do you have career path ideas? What do you guys want to do? What do you want to do? Oh, okay. Science still interests you, though? Yeah. Absolutely. Yep. Anyone else? What they want to do after high school? You don't know? Yeah. You want to be a nurse? So you'll probably be a bio major? Well, yeah, bio major. Yeah.
I want to be a surgeon.
You're going to be a surgeon? Awesome. Research will take you very far in your career path, so keep it up. Yeah.
Where did you go to school?
I went to Albury. Yep, my degree's in biochemistry.
And why did you pick Albury?
So I was between Lebanon Valley and Albury. And Albright was closer to home, so I was able to commute. I paid for college myself, so it's a big money saver. But Albright also has a science program with a 97% placement rate. So out of, I think there were 13 graduating biochemistry majors in my class, 6 of them went to medical school. So their science program is very highly accredited. I learned a lot because I don't know how many people are thinking about college yet, but I wanted to go to school where my class numbers were small. So most of my classes were about this size. My professors knew my name. It was amazing because I could go into their office and I could say, hey, I don't understand this, or hey, I need help with this. And I wasn't just a number. I was able to really get the hands-on experience I needed. So that was also very helpful going to Albright. Any other questions? No other projects? Hello.
Hey.
Long time no see.
Are you just finished?
I just finished.
You guys don't have any questions for me?
No, and they don't have any projects they want to tell me about.
Do you have a molecular project?
That's the one I really
Nothing more?
I know, I don't know.
Is
What
this,
is
you're also pretty scary.
very what are you guys doing mostly? I can touch on more.
Yesterday we talked about like data work. We have real time PCR.
Okay.
I didn't really talk about that with them, but kind of like how we can use DNA sequences to.
What are you guys using for your real-time PCR, instrument-wise?
It's called Chai PCR.
Okay.
It's like a real thing.
Interesting.
I can show it to you.
Yeah, absolutely. Yeah, we use 7500.
Ours is much cheaper.
Yeah. Yeah, I've quickly learned how to most of everything. Okay. Yeah, I don't do a ton of PCR. My group is more cell-based. I do microplasma detection testing, so I do PCR on that. I do ELISAs, I do cell-based immunoassays, and yeah, that's basically what I like most in those two. And then MICO is I was telling them my big project, so I've been on that one since the beginning phase of development, which is pretty cool to see it through. But it's now moving into routine, so.
The one question that I don't know if anyone, like the exact project, one thing that we're trying to narrow down to is we have all these interesting questions, but like what we want to do now is like, what's the one protein, so if let's say you add this drug or this hormone or those chemicals into them, okay, you're hoping to find changes in the cells, but like how do you narrow down and find that one protein in a pathway that you
Right.
Which I know that's like a really broad, that's like something that we can struggle with.
Right. So we don't get a lot of the back history on drugs. A lot of our drugs that come through are literally very beginning phase. So they don't even have names yet. They're just a bunch of letters, and I have no clue what it's being used for. So I didn't tell you guys this. I work for a contract research lab. So we don't develop any drugs ourselves. We do all of the testing for places like Merck, GSK, big names like Samsung, different things like that. So those big pharmaceutical companies send their samples to us and they say, here, do this assay. And we are just basically robots doing their work for them. So we don't get a lot of background information. Our methods don't provide a lot of background information. Of course, they have a scope of what the actual experiment is. So I don't have a lot of ideas in that sense. I know I was talking, I don't know his name, but in the yellow shirt, Jacob. I was talking to him about what he wants to do with like a cell-based assay and doing like the cytotoxicity assay and kind of measuring cell death. But if you wanted to take a study and narrow it down to a specific gene that it kills, like above my level right now. Do you guys do any ELISAs yet?
We've used ELISAs for a couple projects, one was looking for gluten sensitivity.
Okay.
But we can do ELISAs between Western blots if we have we have the ability to look at
I haven't used that since college. And I've never done a Western blot. So our department does Western lots, but that project's currently on hold. I was next in line to be trained on it, and as far as surveys go, everything hits roadblocks and gets put on hold. So
I don't know if this is, it seems like Western Law is kind of like phasing out. There's like other techniques that
Yeah.
are faster.
yeah, I mean, Western lots measure protein, right? So we use a lot of ELISAs for that. Cell-based, I don't know, we do a lot of ELISAs for potency, so we're looking at specific things that way.
So one, like, I don't know if you, are many of you familiar with ELISAs? Are any of you familiar with ELISAs?
I briefly explained, but
So one of the ways that we thought about using ELISAs in the past was like stress. So if someone's really stressed, like there are chemicals that change in the body. One of the big ones is cortisol. So let's say you're interested in stress response due to some, like, there's a lot of studies right now with screens. So could you take like mice or I don't even know if that's in class, but it's cortisol, like expose different groups to different levels of stressors, and then do some kind of test to see does this fly produce a lot of cortisol or some region. You can use an ELISA to do that. So it's just a little test that will tell you this group of cells or this fly is producing lots of this protein. We can do that here, which is cool, but you have to know what protein you're looking for. That's kind of where we are. So, for her, if you don't know what protein is, that's all.
yeah. Yeah, no, I don't. I mean, most of the things we're sent are already past that point, and we're just doing. I work in the routine department, so I don't do a ton of development, but my husband is the only development project I should hold into. But we have a sister department that does all the development work, so we can still have a pharmaceutical company come to us and say, hey, we'd like to do X, Y, Z test, can you develop it for us?
This is an announcement for the SRI students, level 1 and 2 material science classes should report to the ag classroom at this time.
2 is only if you're doing that. You wouldn't have to leave her.
I'll be curious to see if you have someone
Yeah, I think they just asked.
coming from Johnson & Johnson, Medical Affairs.
Okay.
You graduated from Lancaster.
Okay. Okay, so she got me in because I'm looking to break into the big pharma world. But a lot of big pharma companies require experience before they'll let you through the door as an entry level. So
I'll be curious to see his perspective. Like it sounds like
yeah, we're definitely kept away from a lot of the background information just because it's not necessary for our testing. So because I'm not doing any real development work. But just developing skill sets, and I think that was one of the reasons why I decided to go into industry. Is I was stuck between grad school or industry, and I was like, first of all, I'm burnout. I can't do school anymore. And then second of all, I need to develop my skill sets, and my professors in college were like, yeah, go into industry. I'm sure you'll get a leg up. So, and like I was telling them, a lot of people that come to Lancaster labs have very little experience, and they teach every skill set you need.
it's also nice to make more money instead of
It is,
paying money.
yeah, exactly, and studying constantly, so.
We're gonna have to make a new copy. I think. You want me to do it?
I'm about done, unless you guys have questions for me.
Totally up to you.
Yes,
You said there was a dye that helps show which cells are dead and why. What is that called?
Rizathrin, so, I don't know if that's specific to the cell line that we use,
It's all the way up here.
but I can look that up if you like.
How do you spell? REZARIU, or something along those lines. It's a blue dye that will infiltrate dead cells. Okay, thanks.
We do that a little bit. We do like ATP, let's say,
Okay.
Try it right there.
and then,
Oh, yeah.
but that's only for some.
So that'll do, so the Rizathrin, I haven't run this assay in forever, so you're jogging my memory, but, actually,
But we can do this, so, just so you know, like, we have a couple of different ways to measure. Like, if you do cell culture, how many cells are left in your
Yeah.
Oh, I'm sorry, the results are doesn't have living cells. And then it's measured by fluorescence. So I don't know if you guys, do you have any flavors?
Yeah.
Okay, so yeah, as long as your flavor you can read fluorescence. That's another way to.
We do have like fluorescence, it's not very good.
Yeah. Any other questions? I can't disclose like a lot of my testing. That's the other thing that makes it hard is like, I can't give you specific examples of what I'm doing. But
Yeah, if any of you, I can show you everything.
yeah, absolutely.
If anyone wants to sit down and talk about project ideas, always take advantage of them. It's an experience. Even if it doesn't seem like it really quite aligns. I'm gonna run into a couple things.
Okay. Yeah, absolutely. Yes, definitely. I'd love to see. All right.